Good morning, everyone. In today's section, we'll discuss about the 21st chapter, that is biotechnology and genetic engineering. In this section, we'll talk about the application of biotechnology in field of agriculture, in field of medicine, in field of drug delivery system, and how we got genetic engineered crop and genetic engineered organisms, right? So this topic is quite interesting in terms of student, those who are looking for biotechnology field in the future. So without wasting time, let's go to this important topic and the last unit of Cambridge IGCSE biology topic. Okay, and you will expect question from this unit in your paper 1 to paper 4. Paper 1, paper 2, paper 3, paper 4, and you can get database question or graphical question from this in paper 6 also. So let's go ahead. So this topic is primarily focusing on the how to use biotechnology for the human welfare, right? So when it comes to application of biotechnology, there are some microorganisms which are always act as a subject for it. Like we can do all kind of genetic amplification or application or you can say genetic changes in the microorganisms. So we use generally bacteria. So microorganism can be used by humans to produce a variety of things like food, medicine, right? And the most common type of microorganism which we use in this field is bacteria. Because see, there are so many reasons behind why bacteria. Because first of all, bacteria is very small. They have a very small genome, easy to extract, easy to manipulate, and easy to insert the DNA back. And of course, by the process of DNA replication, transcription, and translation, the genes will keep on producing the proteins we required. And always remember the codons are universal, right? So codons are the sequence of mRNA, sequence of bases present on mRNA, right? So generally why we use bacteria? Because they are useful and capable of producing organic complex molecules such as enzymes, hormones, right? We can also produce genetically engineered things. So they're also useful because they grow very fast. They in a very small span of time they can complete the life cycle, right? So they increase exponentially very fast. Biofuels is a again a gift. You can say a gift because in the biofuels we are going to use some kind of or microorganisms which will respire and they will produce ethanol, and ethanol will act as an energy source, right? So we'll talk about this later on in the application section how we are getting. So generally in the aerobic respiration in yeast we get alcohol in the cell, and this alcohol can be used as a power source, as a biofuel, right? The alcohol produced by the fermentation of glucose can be used as a biofuel. How we are going to use it? Let's see. So first, the ethanol produced in this reaction is increasingly being used as a biofuel, right? See, so the reason why we are looking for an alternate energy source, because the natural resources are exhaustible. They can finish at particular time, right? A particular year. And it's easily and rapidly getting exhausted. So if we can move in that kind of energy source where it's abundant, I mean you can continuously produce. So there are some countries which is practically working on biofuel production, like Brazil. So the biofuel is partly replaced petrol in the fuel of car and other vehicles, right? So it's very amazing and a good step in the field of conserving the energy. So plant material is used as a substrate for producing ethanol, right? Because plant have glucose and yeast is going to respire anaerobically and they produces alcohol. And again, you are not going to get only alcohol. We need to purify it, right? Because you're getting a reaction mixture. So the liquid is separated from the remaining solid and other water, right? And then you have to do some separation technique to extract all the alcohol from the solution. So sometimes the waste part of the crop plant, such as some leaves, some dead leaves, some stems, are used, but in other places, crop are grown specifically for this. Like there's a land is occupied by the crop just to grow alcohol. I mean, just to make alcohol, not grow. So in some places, this is causing an concern that there is a less land available for local people to grow food crop needed for survival. So now it's up to you to decide which tank you want to fill, right? Either your stomach or your car vehicle, right? Or your vehicles. So second application where we are using genetic engineering. First is bread making. Second is the bread making. First one was biofuel. Again, here also we are using yeast and they're going to respire anaerobically and it has access to the plenty of sugar, even if oxygen is available. Great, right? So this is, you can say this is an advantage because the same crop which was used to produce alcohol, they are producing CO2 as a byproduct of respiration, anaerobic respiration. So we can use both. So ethanol can be used as a biofuel and carbon dioxide can be used to increase the dough, right? Size of the dough. So the carbon dioxide produced by the yeast during the respiration is caught in the dough, and that's why the dough get rise, right? Fruit juice production. There's a third application. So fruit juice is produced by squeezing the fruit to remove the juice, right? First they chop the fruit, but before squeezing helps to release lots of more juices. But this does not break open all the cells, right? So you need to extract the juice from all the cells. So by adding an enzyme called pectinase enzyme to the chopped fruit, right, there is a chance that juice will be released automatically because enzymes are working. So pectinase works by breaking down chemical called pectin, and that is found inside the plant cell wall. So if you can break the cell wall, of course, the juice will come out. So once pectinate is broken, the cell wall is broken more easily and more juice will be squeezed out from the food. So adding pectinase to the food can help to produce a clear juice and a larger polysaccharide like pectin can make the juice seems cloudy. So if you are having a juice and you are drinking it and it's coming cloudy, that means it is a pectinase. Biological washing powder. Next, fourth one. So generally, the dirt, the stain on the cloth are organic substances such as the oil from the skin, protein from the blood, fat and protein from the food, right? So detergent must contain some kind of, you can say, um, chemical, right, which can remove the stain. So when mixed with the hot water, but it can take a lot of time and effort and a very high temperature to remove the stain entirely from the growth. So that is why what we are doing, we are adding enzymes in the biological powder, which is similarly how they digest, how they act as a digestive enzymes in the body. So using biological washing powder have a several advantage here. So first is it will quickly break down all the large insoluble molecule like fat and protein into the smaller one and make it more dissolve in water. They are effective at low temperature, right? And the third, they can be used to clean delicate fabrics also. which we don't wash at high temperature. Lactose-free milk. So lactose is a disaccharide which is found in milk, and human babies are born with this ability to produce lactase, and that is why they are able to digest it. Lactase. But in certain areas of the world, many people lose the ability to produce lactase as they get older. This means that they can become lactose intolerant and react badly to the lactose in milk and product from the milk. So generally the symptoms what you will find, person having lactose intolerant, they have a nausea, flatulence, and diarrhea because of the upset digestive system is upset due to the lactose. And milk can be made lactose free by just adding lactase enzyme. So lactase enzyme will break down the disaccharide into monosaccharide, galactose and glucose. Penicillin production. This is the first antibiotic produced by Alexander Fleming in 1928. And what he has done, he has notices that the bacteria he had grown, which is left in the petri plate, had been naturally killed by penicillium mold. And the penicillium mold produce a chemical that prevents the infection, right? So the chemical was first isolated and then later on get purified and then named as a penicillin. Afterward, afterward the discovery of penicillin, this method has been developed to produce, you can say, a large amount of antibodies, right? So generally how they do the large production and how they use in fermenter. So first of all, fermenter is a contains a culture. Culture is anything which contains nutrients, water, which helps to grow the microorganism, generally fungus and bacteria. These can then be used for many biotechnological purpose like producing genetically modified bacteria, penicillium mold that produce penicillin. So the advantage of using fermenter is that conditions can be carefully controlled to produce a large number of exactly the right type of microorganisms. So it's very powerful tool. So what are the condition you have to maintain when you are running a fermenter? So first of all, aseptic precautions. Of course, we are dealing with the microorganism and we don't want that unwanted bacteria should grow. So fermenter is first cleaned by a steam to kill all the microorganism and prevent chemical contamination, which ensures only the desired organisms are going to grow. Optimum pH. Of course, we need optimum pH for enzymes to work. So it should not be too much acidic, too much alkaline, then of course enzymes are not going to work. Nutrients. So nutrients are needed for use in respiration to release energy for the cellular growth and also to ensure that microorganisms are able to reproduce. Oxygenation. Oxygen is needed for aerobic respiration to take place. Agitation. So sometimes we have to agitate the mixture right inside because that agitation will mix the uniform distribution of the media, the nutrients, and the bacteria. Optimum temperature, of course, this is very important parameter because of if temperature is not maintained, enzymes are not going to work, and sometimes they get denatured. Now, what is genetic engineering? So, genetic engineering is a changing the genetic material of an organism by either removing it or doing some changes or inserting some new gene from another organism, right? So whenever we form a genetically modified organism, we call them transgenic organism. So the DNA of the organism that now contain DNA from another organism, also known as recombinant, right? So recombination is coming here. There are many examples such as human insulin. There are some crop plants which are insects free. There are some crop plants which are uh, genetically modified in certain genes of bacteria to produce a kind of poison that kills insects. Wow. I mean, if Insect is going to feed the plant, they are not going to survive. So some crop have been genetically modified to produce additional vitamins such as golden rice, right? This rice is having vitamin A, and that could help to prevent deficiency disease in certain part of the world. What is advantage and disadvantage of GM crops? So genetically modified crop means the crop which have mixed form of genes, like lab made concept and the biological core concept, both are mixed. So what is the advantage of using GM crop is reduced use of chemicals such as herbicides and pesticides. So environment will be better and clean. It's a cheaper way, less time consumption for farmers. Increase the yield also. Yes, this is very important. The yield is grown tremendously. Disadvantage, there are many disadvantages. So first is increased cost of seeds. So sometimes seed cost increase. Companies that make the GM seeds change more for them to cover the cost of developing, right? So that is why poor farmer cannot buy it with the large farmers. Increased dependency on certain chemicals such as herbicides that crop are resistant to, often made by the same companies that produce the seed and more expensive to buy. Risk of insert gene being transferred to wild plant. Of course, we are disturbing the genetic base, right? Right. So the GM crop, uh, if it is weeds, also gain the gene that makes resistant, right, to herbicides, reduce biodiversity. Of course, if only one type of crop is there, there is a chance of spread of disease, and if disease is spread, biodiversity will lost. Some researchers show that the plant that have genes inserted into do not grow as well as GM plants. So it's a practical conclusion given by the scientist. Process of genetic engineering process is very easy. First of all, you have to check the gene of interest. When we say gene of interest, it may be anything. Then later on, with the help of restriction enzyme, that gene is cutted, and both ends have sticky ends. A bacterial plasmid is cut in the same restriction enzyme because we don't want that it should be cut somewhere else. So just to make a corresponding sticky ends, we are cutting it. Right? So the plasmid and the isolated genes are joined together. So later on, once your mRNA is formed, it's being joined right on the DNA using ligase enzymes. If two pieces of DNA had matched sticky ends, that means ligase will link them to form a single unbroken molecule of atom, right? So it's quite impressive and good work. The genetically engineered plasmid inserted back to the bacteria, and they will allow to reproduce, right? So after a few months, you'll find a good number of them. The genetically engineered bacteria can be placed in a fermenter. So if you want to do the large scale production, that can also be done, right? So first, how they contain the same genetic code as the organism we are taking the genes from. So its genes are universal. Second point, there are no ethical concerns. There is no problem. And the third is the presence of plasmid, right? This is very important. And with this, we'll stop here and we'll catch you in the next section with the unit 1 and unit 2 because we have not done it. Thank you.